My biggest hellos and biggest hugs to all my KCET and PU2 aspirants out there, my dearest children. I hope you're healthy, you're happy. Well, my name is Jasmine Joy. I teach biology and today is the first session of biology for which I'm very excited. Yes, and I hope you are also excited, at least a little bit excited to see me. Well, we are going to study sexual reproduction in flowering plants and I'm going to tell you what's my plan. And I'm also going to promise you one very important thing. I will be covering each and every line of your NCERT from which questions would be coming. And what should be your promise? Your promise should be to listen to me very carefully. So whenever you're watching the video, it should be just you and me. Nothing else in this whole world should be coming in between. It should be just us, and I'm guaranteeing you, you will be able to understand each and everything. Also, if at all you think that this particular video was helpful for you, what do you have to do? You have to click on the like button, also share it with your friends who are in need. And why is this chapter important, ma'am? Because in PU2, 11 marks came. Okay, 11 marks question comes from this particular chapter. And if you talk about KCET, almost 3 questions comes each year, which is not a small number. You see, the highest number of questions that usually comes is 4 to 5. And in that, if you talk about 3, that's not a small number at all. So this year, it was just 4 number of questions were the highest. So obviously, 3 is also very good number out there. Yeah. Now, if you talk about the chapter, I have segregated it into 4 parts. One particular part would be totally entirely given to the PYQs out there. If you talk about these 3 parts, it would be theory. In the first part, the L1, this lesson 1 session, or I'll call it level 1. Okay, we are going to level up, my dear children. So in the level 1, we are going to cover male and the female reproductive organs of the plant. But what exactly in that? So internal structure of the stamen, that is the male reproductive part, yeah. And then we'll go inside the stamen to see microsporogenesis, and also the male gametophyte, will meet it also. And then detailed internal structure of the pistil would be covered, and then megasporogenesis and the female gametophyte. And we will be solving some questions related to the topic so that everything gets cemented in your brain. Now, one very important thing that I want from you is you should not be opening your NCERT until and unless I ask you to do so. And also, please do not write down anything. You do not need to make the notes. What you have to do is rely on active recall. So what you have to do is listen to me carefully. Towards the end, please ask your brain what all did you cover, and then slowly try to write it down somewhere, or probably at that particular point of time you can open your NCERT, have a read of it and see, oh my god, we covered all of this, and then come down to write your notes. That's the best way. Otherwise, when you're writing and listening to me, you're not concentrating on either of it. And if at all you want the notes, you can join our WhatsApp channel for downloading the notes. I hope I am helping you well. And my dear children, I wish that you secured this position the previous year. So there are a lot of children who were a part of a YouTube team. Okay, they were part of a YouTube channel. They studied from here and also from their respective colleges for sure. Yes, and they were part of different series out there, and they got really good ranks. So it's all because of their hard work and a little bit because of us as well. And we want you to also secure some stupendous ranks like this, and we would be thriving for that. You also have to do so. I'm pretty sure you will do that. Yes. So my dear children, let's make sure the next rank is yours. So over here we have Prajwal with rank 171. So we have to beat Prajwal and all of them who are here. Diksha, Sachin, Kiran, Samiksha, all of them. Okay, so they're equally good. You're also equally good. Try for that. And now, shall we get into the topic? So as I said, we're going to talk about the male and the female reproductive part of the plant, right? And we will begin with the male part. So the male part, what do you call it? The scientific term for the male part, what do you call it? It is known as Androecium, my dear children. Sometimes the name becomes difficult to remember. So what do you call it? You call it stamen as well. So stamen and Androecium is the same thing. And if we talk about the stamen, in your NCERT, the picture is given like this. We'll go ahead and see the actual picture. So over here, you can see there is one stalk-like structure, very long structure, and there is one bulky thing like this. What do you call this? This is known as anther. What do you call this? Anther. And what is this thing? It is known as filament, my dear children. So anther is the place where the pollen grains are being formed. You might have seen some tiny things, right? So these are being formed inside the anther. We'll get into the anther as well. As of now, just have a look at it. Okay, so anther and filament is there. Now, my dear children, if you are asked about the position of the filament, so basically over here, always remember the filament is mostly attached to something like this, which you call as thalamus. which you call as thalamus. And sometimes this particular filament, rather than attaching on the thalamus, it would be there on a petal. So on the top of a petal, you might be seeing a filament coming off like that. So either it can be on the petal or it can be on the thalamus. Now what part of the filament is attached to this? This part is known as the proximal part. Now why am I telling you this? Because I saw a question in NEET and I was kind of very astonished to see that a single line of NCERT can be coming like this also. So proximal end of filament, my dear children, can be attached to thalamus or a petal, and this end would be now known as what? Distal. Proximal means it's near, distal means it's it's away, right? So distal end, it is attached to the anther. So this is about the stamen. Stamen, also known as Androecium, it has two parts. One is the big lobed structure, which is anther, and then there is a stalk-like structure which is known as filament. The filament's proximal end is attached to thalamus or to the petal, distal end obviously to the anther. Done? Now we are going to dive into the anther and see its structure. So over here, structure of anther is something that we are going to discuss. So as you can see over here, there are two very big lobes. So this is one lobe that you can see. Okay, and this is the second lobe that you can see. So can I say that this particular structure, my dear children, is bilobed? Bi means two. So it has two lobes. And now if you see properly, there is a groove-like structure over here. There is a depth over here. So if I try to cut in like this, basically I can divide each lobe into two parts. So this lobe is divided into one part, and the second part here also it is divided into one part and the second part. What do you call these parts as? So this part that you see is known as theca. What do you call it? Theca. So can I say, ma'am, over here the anther is bilobed. Yeah, it has two lobes, and each lobe that I see is having two theca. How many theca? Two theca. So whenever the two term comes, either you use bi or you use di. So it is what? Dithecous in nature. So anther is bilobed, and each lobe that you see has two theca as well. And separating the two theca, what do you have? This structure that you had is known as the longitudinal groove. Longitudinal groove, because it has a depth out there. Now, anther is a four-sided structure consisting of 4 microsporangia. What do you mean by that? So basically, if this anther, I will cut it like this. Suppose I cut it like this and I look from the top. When I look from the top, my dear children, I would be able to see a structure like this. So, bilobed. So this is one lobe, my dear children. This is the second lobe that we are having. So what is this structure? This is one theca, this is second theca. This is one theca and this is the second theca. So basically, if you see, can I say it's four-sided? 1, 2, 3, 4. It looks four-sided. It's tetragonal. Tetragonal means four-sided. Okay, tetragonal structure consisting of what? 4 microsporangia. What is that? So basically, inside this, they have some structure like this, and they have given it a name, that is microsporangia. Okay, sporangia. What is it? We are gonna talk about that as well. Well, I can just roughly say that it is a sac of pollen grain. This is where the pollen grains are made. Always remember, microsporangia is not the pollen grain itself. It is a sac of pollen grain. It has all the pollen grains inside it. It is where the pollen grains are being formed. So microsporangia can also be known as the pollen sac. The microsporangia develop further and becomes what? The pollen sac later on. So my dear children, anther which is there, it is bilobed. Each lobe that you see has two theca, so it is dithecous as well. Now if you cut from the top and you look from the top, it looks like a four-sided structure, and each side of it has one microsporangia each. This microsporangia that you see in future, it becomes what? The pollen sac. Clear? Yes? If it's clear, give me a thumbs up. Give me a thumbs up or put up a like. In the there's a like button, right? You can click on that as well. Either way, you have to tell me, either in the comments or somewhere. I just want to know that you are able to understand. Now we are going to dig deep into the microsporangia and its walls. So my dear children, I have this picture, very pretty picture out there, but I would like to make one not so pretty picture, because that's how you might be able to understand. So over here, I have the two theca. Similarly over here also I have it. I'm not making the entire thing over there. So okay, if you want, I can make it as well. So This is one lobe, this is the second lobe, this is one theca, this is second theca, third theca, fourth theca. Okay, so basically in one lobe you have two theca, here also you have two theca, that's why it's dithecous. Now my dear children, so basically I told there is microsporangia, and the structure of microsporangia is kind of difficult. Oh sorry, not difficult, it's different. Okay, sorry for using the term. So over here, internally, this is where the actual go down is, where the pollen grains are made. Okay, so what do you call this? This is known as sporogenous tissue. This is the go down where the pollen grains are made. So it's a tissue, that means it has so many cells, my dear children. Yes. Are these cells the pollen grains? No. These cells are the ones that would be making the pollen grains. Now, leaving that over here, I'll go outside. So outside over here, I have one layer. This particular layer that you see, it is known as epidermis. It is known as epidermis, okay? And now if you go within, there is one more layer, and this one more layer is known as endothecium. What do you call it? Endothecium. So first epidermis, second endothecium, and then sporogenous tissue? No. In between that, you have two more layers. So over here, the sporogenous tissue which was there, which was going to work hard and make the pollen grains, it kind of feels lonely. And to nourish it, to say that everything is fine, it has one extra layer out over here, my dear children. And this layer is the only best friend of sporogenous tissue. It is known as tapetum. What do you call it? Tapetum. And tapetum kind of loves the sporogenous tissue. It provides nourishment and everything out there. Now in between this and this you see space. Can it be empty like that? No. There will be certain cells out there, a layer out there. And what do you call this particular layer as? There is a layer out there, and this particular layer, since it is in the middle of the tapetum as well as the other thing, you call it as the middle layer. So this is a third layer, and this would be the fourth layer, and inside that you have the sporogenous tissue, my dear children. Okay, so ma'am, you said that tapetum is the one that nourishes the sporogenous tissue out there, right? What about the other guys? They are just there, idly standing over there? No. They are there for protection. Okay, they will be protecting, and also the pollen grains are going to be made over here, but they have to move out, right? Then only these pollen grains can go somewhere on the stigma of some other plant and make the fertilization happen. Sexual reproduction has to happen, right? So basically, to help the anther break open, okay, to help the anther break open and release the pollen grains, who would be helping? These three layers out there, epidermis, middle layer and endothecium. So I'll say that they have two functions. One is protection, and the second one is to help in dehiscence. Dehiscence is nothing but basically breakdown of the anther to release the pollen grains. And tapetum, if you talk about it, as I told you, it's there for nourishment. And since it's there for nourishment, it also should be having something to nourish. Okay, for example, your mother and your father would be giving you food. So if at all, only they can help you only if they have the food. Right, they should be having abundant stock of food, then only they can feed you with that food, right? Similarly, the tapetum also, it has very dense cytoplasm. Okay, the cells of it has dense cytoplasm, and it has that much cytoplasm, but it forgets that, okay, the cell division has to be completed. So sometimes what happens is that usually when there is one cell, there is one nucleus, and then when this particular cell divides, you get two cells and you have one nucleus each, right? But over here in the case of the tapetum, my dear children, basically divides, but what happens? The nucleus also divides further, but it forgets to divide completely. So there would be two nucleus in one particular cell. There would be two nucleus in one particular cell. So what do you call this division of nucleus? Karyokinesis. Karyokinesis. So karyokinesis has taken place. But what about the cell division? Has a cell totally divided? No. So I'll say no cytokinesis. Cytokinesis has not been completed, but karyokinesis has been completed. That's an extended version of it. If at all you do not want to remember it now, it's totally fine as well. But as of now, you remember that it would be having more than one nucleus because it forgot to divide because it was nourishing. So I'll say it is binucleate. It at least has two nucleuses. Okay, so dense cytoplasm and pinucleate structure. Inside that you have sporogenous tissue. Each cell of the sporogenous tissue can give rise to pollen grains, my dear children. Okay, so each cell of this sporogenous tissue, I'll call it PMC. What do you mean by PMC? Pollen mother cell. It's a mother to the pollen grain out there. You also have one more name for the same thing. It is also known as microspore mother cell. So, ma'am, it's giving rise to pollen as well as microspore? No. Actually, it just gives rise to microspore. This microspore is the one that would be further developing into what? The pollen grains out there. That's why it's used simultaneously in the same manner. So, microspore mother cell, MMC or PMC would be totally fine. Each cell of it are like that. Now, this is a very important thing, and usually this question is being asked, what are the layers? Put down the name of the layers out there. What do they do? Okay, which of them are involved in protection and dehiscence? Which of them is there for nourishment and things like that? So you have to remember the name. And now I'm very excited to share with you one very important thing. Okay, so there's a fun way to remember it. I tried to download it somewhere. So there's a song, you can remember it by this. So epidermis stands out white, endothecium just inside, middle layer walks away, tapetum comes to save the day. So that's the mnemonic that I have. I'll say once more. Epidermis stands out wide, yes, and endothecium just inside, middle layer walks away, and tapetum comes to save the day because it is there for nourishment. So this is something that you can, if you want to remember, you can remember it this way, because this particular thing is very, very super important that you can say. Okay, so that's the thing. And now I hope you like the rhyme as well. I am trying to make it as interesting as possible. Some people even think, how can ma'am be, ma'am sometimes is so over exaggerating things, but that's my way to teach you guys so that it remembers. You have to do a little bit of drama at times to have that attention, because you know what, the attention span of normal human beings is very, very less, and to keep it intact you have to do something. Yes, so wall layers, 4 layers, epidermis, endothecium, middle layer and the tapetum. Functions: outer 3 layers protection and help in anther dehiscence. Tapetum, the innermost one, nourishes the developing pollen grains. The tapetal cells, or basically tapetal cells out there, it has dense cytoplasm. Often it is binucleate due to mitosis without cytokinesis. Mitosis has happened, okay, but the cytokinesis, the cell has not divided completely. Now, sporogenous tissue found in the center later forms the microspores or the pollen grains. It's again not the same. Microspores would be giving rise to the pollen grains. So, ma'am, you're telling from a long time pollen grains would be made. Until now, it's not made? No, it's not made. So, now we're going to talk about how pollen grains are going to be made. And that is all about the microsporogenesis, the process of formation of microspores, my dear children. So just a second, I have touched somewhere. Yes, now it's fine. Okay, so let's see that. So over here, I told you, inside the sporogenous tissue, each cell out there is going to act like a microspore mother cell or a pollen mother cell. Now, what type of division do they undergo? Have you heard about meiosis? So, whenever you want to form the gametes or cells that would be forming gametes later on, okay, the cells undergo a type of cell division which is known as meiosis, about which you might have studied in your PU1. Okay, so when you start with meiosis, you would be having a single cell out there. Meiosis happens in 2 stages. There will be first division where you get 2 cells. Again, there will be division. You will get 4 cells at the end. So, from one meiosis division, you get 4 cells. But there is one very important thing. Why do I say that the gametes show this particular thing? What is there so unique in gametes, and what's there so unique with meiosis as well? Is it just because you get 4 cells? No, my dear children. So, suppose this particular cell had 2n number of chromosome, double number of chromosome. Okay, so by the first round of meiosis, this particular cell, it becomes half. So it gives half to it and half to it as well. So it becomes n. Here also it would be n. So you see, the number of chromosome, it has become half over here. Similarly over here, when it divides, do you think it will become more half? No, my dear children, it will stay the same. What do you call this as? You call this as diploid. Diploid as in you have double the number of chromosomes, right? And this one where you have half of it, you call it haploid. So basically by the end of meiosis, you get cells that has half the number of chromosomes. Why? Because there is male gamete with half the number of chromosome, there would be female gamete with half the number of chromosome, both of them would be coming together, and now n plus n would be giving you 2n diploid cell, a normal cell out there. So that's the reason why the gametic cells, they undergo meiosis. Always remember that. So now over here, let's come back to the topic. So over here you have the microspore mother cell. What is it going to undergo? Meiosis. And meiosis happens in two stages. So first meiosis happens, my dear children, from 2n it has become n and n. Okay. And now there is second round of meiosis that happens. Over here you get this cell divides into 2, this cell also divides into 2, all of them having n number of chromosomes. Now it is not getting reduced. This whole cycle of meiosis is one meiosis cycle. Always remember it happens in 2 stages, but it is just one meiosis. And the small junu cells that you're getting, what do you call it? You call it microspores. You call it microspores, my dear children. Ma'am, you said you're going to tell how pollen grains are being formed. So over here, these microspores that you see, okay, there is some unique thing about them. I'll just erase it because something else is also going to come over here. The unique thing is basically they do not stand away like this. They usually, when they are formed, they are a form of tetrad. Okay, tetrad as in 4 of them are going to stay very close together. So you call this microspore tetrad. When they are undergoing division, at the end you get something like this. So later what happens, this microspore tetrad, no, it will move away from each other and they will be forming 4 cells out there, and these 4 cells all together, you would be now calling them as what? Pollen grains. You would be now calling them as pollen grains. Until now you were not supposed to call it as pollen grains. Okay, got it? So sporogenous tissue each can form a microspore tetrad. Microsporogenesis is what? Meiosis in the pollen mother cell or the microspore mother cell, which produces microspores. Microspore tetrad is being formed, initially grouped in 4s, but later they will be separating. When does this happen? When anther is matured. So anther would dehydrate, it will lose all of its water. The microspores will dissociate when there is no water and develop into what? The pollen grains. Later on, there will be release of pollen grains. Thousands of pollen grains are released when the anther dehydrates. When the anther will be dehiscing, when it will be breaking down, all of it would be moving out as well. So this is about microsporogenesis. So starting with diploid cell, you get haploid microspores, which later move away from each other, form the pollen grains. Okay, now ma'am, is the pollen grain the gamete itself? Is the pollen grain the gamete itself? No. Pollen grain would be having the gamete. So the one that has the gamete, you have a separate name for it. You call it the male gametophyte. So over here, all about the male gametophyte, we are going to talk about the pollen grain, because pollen grain is the one that would be having the male. It's not the male gamete. It would be having the male gamete, which is why it is known as the male gametophyte. Okay, so you got microspore, which later transformed into pollen grain. And the pollen grain, my dear children, what happens in the pollen grain? It undergoes a process that is known as mitosis. Okay, so inside the cell, inside the microspore, you had just one nucleus. This was the condition. You had one nucleus out there, the haploid nucleus out there. So it will undergo mitosis. Basically what happens? You should be getting 2 cells like this, each with its own nucleus, right? But over here, in the case of pollen grains, things are quite different. When the cell division takes place, both the cells are here itself. So when the cell which is there, it divides, now you get 2 cells, which looks like this. So one cell with a bigger nucleus, small cell with a smaller nucleus inside this particular cell itself. So they do not separate out, it's within the same cell itself. So they undergo mitosis and it looks something like this. Okay. So basically, the bigger cell out there, I'll repeat it again because I know it's confusing. So instead of dividing into two cells completely, moving away from each other, cell division takes place within the cell only, and now the cells are here itself. The bigger cell is known as the vegetative cell. What do you call it? Vegetative cell. And the smaller one, you call this generative cell. Why do you call this as generative cell? Because this is the one that is again going to undergo mitosis. Only that one, okay? It is again going to undergo mitosis and give rise to two male gametes. So inside one pollen grain, which is the male gametophyte, you first underwent one mitosis, you got one vegetative cell, one generative cell. This generative cell again divides and gives rise to what? Two gametes out there. It will be giving rise to two gametes out there. I know it's kind of confusing, but you will get a hang of it. Once you go through your NCERT as well, you'll be like, oh, okay, like that. Okay. So my dear children, the male gametophyte pollen grains represent the male reproductive units. Observation, it's there in variety of size, shape, color and design. The size is spherical, 25 to 50 micrometers in diameter. This number is also very much important. Okay. What is the diameter of this? 25 to 50 micrometer. That means it's very small. Okay, now if you talk about the structure of the pollen grain, my dear children, you have to remember that as well. So over here, this was one cell which divided into two. So obviously there is one bigger cell like this, and inside this cell only you have one smaller cell like this. So this is the smaller cell. This cell that you see, it is known as vegetative. It is not going to do anything. Okay, I'm gonna tell you what it actually does. So this is vegetative cell, and this one is the generative cell, because this is the one that will generate the gametes. Okay. Now obviously this is a cell, so it will be having plasma membrane. So this is the plasma membrane that it has. I'm not writing everything, but I hope you get it. Now outside it you have one layer. This one layer is continuous. And what do you call this particular layer as? This is known as intine. This is known as intine, okay? And intine, my dear children, it is made up of two very important things. One is cellulose and the other one is pectin. Okay, cellulose and pectin. Now, ma'am, that's it? There is only one layer? No. These pollen grains, once they are released, they go through wind, water, a lot of things. So they have to stay safe, which is why they have three layers. One layer is plasma membrane, the other layer is intine, and then you have one more layer, which is not continuous layer, which is not a continuous layer. And what do you call this layer as? This layer is known as exine, the external layer out there. And as you saw, it is not continuous as well. And you know what? One very important fact about it, this is made up of the toughest thing in the universe, which is known as sporopollenin. What do you call it? Sporopollenin, my dear children. It is the one that would be making this particular pollen grain safe. On the sporopollen, my dear children, if you put alkali, if you put acid, still it will be all safe, which is why I'll say it is the toughest thing out there. And it is very important for you to learn this, because so many questions comes off from this particular topic as well. Now, wherever sporopollen is not there, that's where you get small gaps. And what do you call these gaps as? You will call these gaps as the germ pore. The germ pore, my dear children. But what exactly do you need? Or obviously for exchange of moisture and everything out there. Apart from that, so these pollen grains, they obviously have to go land on the female reproductive part, right? And then the pollen grains have to transfer the male gametes, which this generative cell will be making out there. This generative cell will be dividing into two. You get two male gametes out there. So these are the ones that has to be transferred, right? So what happens is that it's not easy for getting it transferred. First thing that happens is this vegetative cell. Now I'm going to talk about what is its role. This vegetative cell, you also have another name for it. You also call it tube cell. Why, ma'am? Why do you call it tube? It does not look like a tube. So basically, this is the one that will slowly bulge out like this. Slowly bulge out like this and form a tube-like structure, which is known as the pollen tube. So when basically, you know, the female reproductive part, there is stigma, there is style. We'll be talking about it in the next session, okay? The stigma style. So over here in the ovary, you have the female gamete, right? And over here, the pollen grains would be coming and landing, and they cannot just get inside. Instead, they would be making a tube-like structure, and they will be sending their male gametes through this. So the pollen tube is very much important for that. The generative cell will be dividing, and the pollen grains would be moving down through this particular tube. And since the vegetative cell is there for making this pollen tube, you also call it tube cell. Apart from this, there is one more function for this vegetative cell, which is very densely, which has very dense cytoplasm. It is there for providing nourishment. It is also there for providing nourishment to this guy, that is the generative cell, because the generative cell will be dividing into two, right? Always remember that. And as you can see over here, the picture that I made, there is one very important feature. I'm going to tell about the feature of the vegetative cell and the generative cell, okay? So vegetative cell, it has dense cytoplasm, agreed? Dense cytoplasm, and apart from that, if you see its nucleus, it is irregularly shaped. It would be what? Irregularly shaped. Now, if we talk about the generative cell, that is the one that generates the gametes, over here the cell will be small, and if we talk about the nucleus, it is spindle shaped. Almost the cell is actually spindle shaped. Spindle shaped cell and nucleus. Nucleus is kind of elongated but still small out there, and this is the one that will give rise to the male gametes. I hope this is clear. So in time, I'll just sum it up. Okay, there are two cells over here. The pollen grains undergoes mitosis, has two cells. One is the generative cell, the other one is the vegetative cell. Okay, where is the generative cell? Inside the cytoplasm of the vegetative cell out there. Now, they have their own plasma membrane, but outside that you have an intine. Intine is made up of cellulose and pectin. Outside that you have a discontinuous layer which you call as exine. Exine is made up of the toughest material out there, which is sporopollen, my dear children. And there are certain spaces where it is not there. You call them as germ pores, through which the pollen tube is formed. Who helps in the formation of pollen tube? It is the tube cell of the vegetative cell. Ma'am talked about vegetative cell. So basically it has dense cytoplasm, irregularly shaped nucleus. It is there for nourishment of the generative cell. The generative cell that you see, it again undergoes mitosis, giving rise to two male gametes. And if we talk about the cell structure, it is spindle shaped, and the nucleus is also very kind of small and elongated structure that you have. Done. And if we talk about the diameter, it is 25 to 50 micrometers. If you want, you can take a screenshot of this, because I have not added this page up there, so it might go off. Okay, done. Now so where are we? We are here. Chalo. So exine is the outer layer made up of sporopollen, highly resistant material, has germ pores. I told you. Intine is the inner layer made up of cellulose and pectin. Cytoplasm, it is enclosed by the plasma membrane. Cell types in the pollen. So there is vegetative cell. It is larger. It stores food and has an irregular nucleus. The generative cell. So smaller, spindle shaped, floats in the vegetative cell. Now the pollen shedding. That means the pollen grains, when they go off like that. In how many cell stages do they shed? That means how many cells are there within. So either it can be in the two cell stage. That means there is one generative cell and one vegetative cell. Almost 60% of the plants, the majority of the plants, they go in the two celled stage only. Okay, the generative cell has not divided yet. But in the rest of the 40% of the plants, they shed at the three-celled stage. That means the generative cell has already undergone its division to form the two male gametes out there. So 60% of the plants in two-celled stage, 40% of the plants in 3-celled stage. Always remember that pollen grain is not the male gamete, it has the male gamete, which is why it is known as the male gametophyte. Now, for you to have a real-time picture of how it actually looks, it looks something like this, my dear children. So this is the tube cell that you have, and this small little structure that you see over here, this chotu cell that you see over here, is the generative cell. Okay, and always remember, whenever you're talking about sexual reproduction, you're talking about the flowering plants out there. Okay, and they are known as angiosperms. So 60% of the plants are the flowering plants, that is the angiosperm. 40% of the plants are also angiosperms. So this is all about angiosperms. Remember that, the flowering ones. Okay, done. So we are officially done with the structure of the stamen. the microsporogenesis, the male gametophyte as well, and now we'll talk a bit about the pollen grains. Okay, so you know what, pollen grains, apart from sexual reproduction in plants, they have many other functions. Some people eat it, and it helps in the performance boost up. Okay, for example, there are athletes who basically take pollen grains because it enhances their stamina out there. Okay, there are different pollen products, syrups, tablets and things like that, which people use in Western countries. It is because it is obviously highly nutritious, because you are making the male gamete, and the male gamete, my dear, it has to be healthy and things like that. So obviously whatever cells, whatever things are up there, it will be very, very nutritious for some people as well. But whereas some other people, they are very, very allergic to this, so which is why it is said that pollen can cause allergies. It causes asthma, it causes bronchitis, it causes respiratory disorders in some people. And there is one more very bad thing about the pollen grain over here. So that is Parthenium. I'm pretty sure you have seen this particular plant. Okay, I didn't enhance the color too much, otherwise it would have been so dark over here because of the contrast. Or if at all, you can please go towards Google later on and just put down Parthenium and see how does it look. I'm pretty sure you have seen it. So Parthenium, you also call it a carrot grass. It is an invasive weed, as in we brought it from somewhere. It is a weed. Okay, it is a weed that takes away nutrition from the actual plants, and it is the one that actually causes severe pollen allergies in India as well. So people, when it brought it, they brought it probably because of the beautiful flowers and cute little things like that, but it was causing trouble to the native plants out there. And apart from that, now it's causing trouble to us because it causes pollen allergies. So pollen grain can be acting as an allergen that causes allergies. It is used as tablets and syrups in Western countries, and sometimes it can be used as performance booster in the case of athletes as well. And now there is one more very important thing about the pollen grain, that is the pollen viability. What do you mean by viability? Basically, the pollen grain which is there, when it goes and lands, my dear children, it has to make sure of one thing, that is the pollen tube will be forming and the male gamete should be going inside and fusing with the female gametes out there. So this process is known as the viability. Okay, viability is basically basically this capacity to germinate, to form the pollen tube, to give the male gametes. All of this is known as the viability. So pollen must land on stigma before losing its viability, having the capacity to live, grow and germinate or develop for fertilization to happen. Now, depending on certain plants, you have different types of viability period. For example, there are plants who say that once the pollen is released, within 30 minutes, I am going to lose the viability. After that, this particular pollen grain would not be of any use. Who says that? The one that we eat, that is rice and wheat, my dear children. So rice and wheat loses viability within 30 minutes. So within 30 minutes of pollination has to happen, and later on, obviously, so the pollination has to happen. It has to land on the stigma of the correct plant out there. Otherwise, it might be not of any use. Okay, so it has very short-lived pollen viability. Now, there are plants which has long-lived pollen viability as well. They would be viable for months. Who are those plants? So I have given you a picture over here because sometimes it becomes difficult to remember the family's name. So rose belongs to Rosaceae. Okay, over here you have a legume, that is a bean. Okay, and bean belongs to Leguminosae. Okay, and over here you have chili or potato, everything, they belong to Solanaceae. So Rosaceae, Leguminosae and Solanaceae. Okay, I feel I am talking Spanish, but yeah, so these are the families which basically whatever they give out, they have the pollen viability for months. Okay, so these are examples that you have to remember. Now, suppose I have the pollen grains of a plant that is soon dying, it's soon getting extinct. What can I do? Basically, I can store the pollen grain somewhere and make sure that whenever I want to germinate it, I will do it in my laboratory conditions and stuff like that, so that the plant is not extinct. So there is a way to store the pollen, my dear children. How? The pollen can be stored in liquid nitrogen. Okay, at minus 196 degrees Celsius. Over here I have almost 20 degrees Celsius. I am freezing over here. Okay, now you imagine minus 196 degrees Celsius. In that, if you keep that particular pollen grain, obviously it would be also freezing, but it will be completely safe. Okay, safe to use later on. So liquid nitrogen is a thing where we put it, okay, and at this particular temperature as well, where it won't be losing its viability for years. Now, Where do you keep it? In the pollen banks. Why do you keep it? For the crop breeding programs, okay, for breeding the crops, different type of crops are there, and yes, things like that. Okay, so this is a general thing that you should be knowing about pollen grain, which is there in your NCERT, and with that I'm pretty sure we have completed the male part of the plant really well. Now you can open your NCERT if you want. Okay, you can pause this video and go through your NCERT once, or if you think that you have already understood everything, I'll recommend you to listen to me more and complete the female part, and then you can come ahead and basically read the whole thing together. That also works. Either way it works. And my dear children, if at all you are understanding what I'm saying, you have to put it down in the comment section. Say that, ma'am, I'm understanding, or ma'am, this is the question that I'm having, or whatever it is, and also click on the like button. Okay, and also share it with your friends who are in need. Any CBSE board, PU board, whoever are there, whoever is in need, help them out as well. Okay, so just taking a 1 minute break, I'll be coming back. Okay, let's go ahead with the female reproductive part, which is known as Gynoecium. What is the male reproductive part known as? The Androecium. Remember that. So now, since the Androecium has a different name, Stamen, like similarly, Gynoecium also has a different name for it, and it is known as Pistil. It is known as Pistil, or it is also known as carpel, my dearest little children. And I'm going to tell you, this is going to be the fun part of this particular video, because it has a lot of things. You might be astonished to see all of this. I am going to show you certain pictures also. It will be really nice. So the picture that is given in your NCERT, it looks somewhere like this. I'll tell you how it looks. So this is the stigma that you have, and this is the style that you have. And my dear children, this is the ovary that you have. So you have three parts of the female reproductive part, or the pistil, or the carpel, or the gynoecium. One is the stigma, right there. This long thing is style, and this thing that you see is the ovary. Inside the ovary, you have the cute little thing that is known as ovule. What is ovule, my dear children? This is where the female gametes are going to be formed. It is not the female gamete itself. It is where it is going to be formed. So, ma'am, is it the female gametophyte? No, it is not the female gametophyte also. You can compare it with what you call the sporogenous tissue, okay, or you can compare it with the anther. That will be the best thing. So the anther has the sporogenous tissue. The sporogenous tissue would be forming the pollen grains. The pollen grains forms the gametes, right? Similarly over here, the ovule is a structure that would be having something like sporogenous tissue. That sporogenous tissue would be forming the female gametophyte. That female gametophyte would be forming the gametes out there. Okay, so this is general structure, but how does it actually look? Obviously, it varies in different things. But over here, you have the stigma, which is, as you can see, glossy. Okay, so it is a sticky thing on which the pollen grains would be coming and sticking up. Over here, you have a long style, and over here, you have little ovary. Inside the ovary, my dear children, you would be having ovule, which is not mentioned over here. And this particular thing is thalamus, on which it rests. Now, so stigma, landing platform for pollen. Style is the slender stalk. Below the stigma, ovary is the basal swollen part containing the ovarian cavity. So there is a cavity. This particular space, just a second. This particular space that you see over here, it's nothing but a cavity, okay, and that is known as the ovarian cavity, or you also call it as locule. What do you call it? The cavity is known as locule, and in that, who is staying inside? It is the ovule, my dear children. So ovule which is there, it is known as the Megasporangium. Inside the anther, you had Microsporangium, right? Similarly, over here you have the Megasporangium, which is known as the ovule. And I'll tell you one very important thing that you have to remember. When you say a fruit is being formed, okay, when you say a fruit is being formed, basically a fruit is a result of fertilization. That means male and the female gamete has fused, okay, and when it fuses, my dear children, this ovule that you see becomes the seed. It becomes the seed. What about the ovary? It becomes the fruit. Okay, so in plants like papaya, where you have so many seeds, what do you understand from that? That means it would be having so many ovules as well. Okay, example, mango. If you talk about mango, it just has one seed. That means it would be having just one ovule within it. So that's a different thing. I'll be showing you pictures related to that, okay, for your understanding. But this is the general idea of the pistil, or the carpel, or the gynoecium as well. Now going ahead, there are different types of pistil. Okay, not different types of pistil. Basically, suppose there is only one pistil out there. one stigma, one style, one ovary. You call this condition as monocarpillary condition. That means mono means one, single. So monocarpillary condition, that means single pistil is there. Okay, now there is another condition that is known as multicarpillary condition. That means multiple carpels or multiple pistils are there, my dear children. Now in this case, when multiple pistils are there, either all of them can be fused like this, they can be fused with having a single ovary, or they can be separate but still together in one particular thing. So this condition where they are all fused, you call them as syncarpous condition. Syncarpous, as in all the carpels are together. Apocarpous condition means basically you have so many carpels out there, but all of them are separate. All of them are separate. Okay, that is apocarpous condition. So syncarpous condition, it is usually seen in papaver. Papaver, my dear children, you would be studying this in human health and diseases. There's a drug that is known as opium or heroin. Okay, so this plant where you get this drug is known as the papaver somniferum, and this is the plant out here. This is the picture given in your NCERT about papaver. I'll be showing you the exact picture, how does it look and things like that. So over here you see, what is this? This is the stigma. This is a stigma of different what do you call, carpels out there, but all of them, my dear children, would be having just one ovary out there because all of it is fused. This is syncarpous condition. And what is this particular thing that you see? These are the pollen grains, the anther, the stamen out there. So they have multiple stamens as well. Okay, so over here syncarpous condition, because as you can see the stigma is there, but all of them are fused together to have a single ovary inside. So this is about the papaver. Okay, and if you talk about the apocarpous condition, it is where you have multiple pistils, but yes, you have it. So this is a condition out there. Where can you see it? In this particular plant, or the flower or the plant, basically it is known as Michelia. What do you call it? Michelia. In Malayalam you say Chembagapu. Okay, in some other language also it is known as Champak and things like that. In Hindi it is known as Champak. It smells so good. So if you open the Champak flower, my dear children, what happens is that you would be seeing some structure like this, where these are the stamen out there, and these structure that you see are the carpels out there, and at the top, if I zoom it in, this structure is the stigma and the style region, and they would be having separate ovary out there. So they have so many carpels. That is why they are coming under multicarpillary condition, and why they come under apocarpous condition, because all of them are separate. Okay, so this is an example given in an NCERT that you have to remember. If at all you forget, you might lose your marks here and there. I don't want you to do that, and this is going to help you in your NEET examination as well. Okay, done. So we have had a basic idea about that. Now we are going to dive deep into the ovule, the structure of the ovule, the structure of the ovary and things like that. Just give me a second. Yes. Okay, so structure of Megasporangium, which you also call as ovule, my dear children. How does it exactly look? So I am going to make a separate picture for that. So you have got your style right over here. And this is the mighty big ovary out there. Yes, even though it does not look good, please don't mind. You have your ovule over here. I am just making one single ovule. Suppose this is mango plant. Okay, what exactly happens is, this is the locule or ovarian cavity, right? Now, somewhere around here, you might be having a structure that is known as placenta. Ma'am, why do you need placenta? Placenta is there in human beings. What does it do in human beings? It provides nourishment, right? Similarly, here also it provides nourishment to the ovule out there. Okay, now this particular structure that connects the placenta with the ovule, it is known as funicle. What do you call it? Funicle. It's very funny. Okay, it's just there. It's funicle. Okay, funny, you can remember funny way, or whatever you want to remember, you can remember that way. Anyways, so it is attaching at a point, and this particular point is known as hilum. So basically, this placenta just wanted to say hi to the ovule, and in a funny way, which is why you have funicle in between. I know that's a very bad way to remember, but that would be helping you. Sometimes bad ways also help you well. So now, my dear children, the ovule says that I'm feeling very cold inside. I need 2 sweaters. So there is one sweater out there like this. Okay, this is known as the inner integument or the inner layer out there, and there is one more sweater out there that is known as the outer integument. meant. Okay, ma'am, why are you not covering? Array, it needs space to breathe and all, right? So this particular pit that you are seeing, that open pore that you see, you call it as micropyle. What do you call it? Micropyle. And this region is known as the micropylar region. This region is known as the micropylar region. Now, wherever you have the micropyle, the opposite side of it will be known as the chalazal region. The chalazal region. Okay, so just to give you an overview, there is ovary, there is placenta there for nourishment. The placenta wants to say hi. So this point of attachment is known as hilum, and it does it in a funny way. So you have the funicle. Okay, the ovule still feels very cold inside it because it's like very cold. I don't know why. So it has two sweaters, inner integument and the outer integument. One way we have left it for passing of air and stuff like that. You have the micropyle region for that. Okay, micropylar region. The opposite end of the micropyle region would be known as the chalazal end. Now, what is there inside the ovule? That is also very important. So there are cells around over here. Okay. And these cells are known as the cells of the nucellus. It's not nucleus, always remember that. It is just cells of the nucellus. Just like any random cell out there in the plant, they are all diploid in nature. They are all what? Diploid in nature. And in between that, there will be one mighty king, no, not king, it's queen. So one queen cell out there, which is going to be known as the megaspore mother cell. Megaspore mother cell. Just like how you had in your sporogenous tissue, just remember what was happening. So this is the ovule. Basically, you can think ovule to be the anther, or if you do not want to think like that, just think of it as the megasporangium. Okay, it is the megasporangium out there. Inside the megasporangium, which is there, you have so many small cells. It is known as nucellus. One cell among them, it would be known as the megaspore mother cell. Always remember, in the case of males, in the case of the male, male plant, all the cells were participating, but over here only one cell is going to participate in the formation of the megaspore out there. Okay, so that is this particular megaspore mother cell, which is also diploid, and it's a part of the nucellus cells as well. So this is the general structure that you have to remember about the megasporangium. Now, I want you to have your attention over here. Suppose the placenta is somewhere over here and the funicle is here, the hilum is here. Will you still call this region as the chalazal region? Yes, you will still call this particular region as the chalazal region. The opposite side of the micropyle would be always chalazal region. And whenever you have a condition like this where basically the placenta is towards the opposite end and things like that, you will call this ovule as anatropous ovule. What do you call it? Anatropous ovule, as in it is kind of twisted out there. You call them anatropous ovule. Got it? Yes. So please have that understanding as well. Opposite side of micropyle is always going to be chalazal. Now we are going to read about it. So ovules is known as microsporangia. It arises from the placenta inside the ovary. Placenta is located inside the ovarian cavity. I told you, inside the locule. Funicle is the stalk attaching ovule to the placenta. Hilum is a junction between the funicle and the ovule body. There are integuments, one or two protective layers leaving a small opening that is known as micropyle. The small opening at the tip is for the pollen entry and also for the exchange of gases and stuff like that. Chalaza is the opposite end of the micropyle, the base of the ovule. The nucellus, mass of cells with reserve food material. So they are actually there for the food, reserve food material. So this is general structure that you are having. Now we are going to see what happens with the megaspore mother cell. It is going to undergo something which is known as megasporogenesis. Okay, it is going to undergo something that is known as megasporogenesis, and you might see something over here. We'll come to this back again. Okay. So megasporogenesis is the formation of megaspore, where what exactly happens is that same thing, just like how in the case of male cells, the microspore mother cell was dividing, here also it will be dividing, undergoing division. First meiosis, where it becomes N and N. Here also second meiosis, N, 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 N. At the end, you get 4 cells out there. What do you call these 4 cells as? You call them as megaspores, not microspores. You will call them as megaspores out there. Okay. Now, there is a twist in this story. In them, in the case of males, all of them were forming the pollen grains. But over here, three little kids are gonna degenerate, my dear children. Only one of them would remain functional. It is the one that will be forming the female gametophyte, the one that will again form the female gamete out there. So there is only one functional megaspore that will go ahead for doing all the process. Rest all of them would be going off. Okay, so always remember there one microspore mother cell gives rise to 4 pollen grains, but over here only one megaspore mother cell will only give rise to one functional megaspore, and this one functional megaspore will only give rise to one embryo sac. So embryo sac is the name of the female gametophyte. Okay, embryo sac is the name of what? The female gametophyte. The female gametes are going to be formed. And where is it all happening? Inside the ovule. Inside the ovule, you had the nuclear cells, one cell divided and formed all this thing. Rest of the nuclear cells are still there surrounding it. Remember that within the ovule. So your story is something, it looks like this. So over here, you have the ovule, the nuclear cell are still surrounding it. Okay, one cell divided and you have got one functional megaspore. This one functional megaspore which is there will now be very big and form what? The female gametophyte, where the female gametes are going to be formed. Okay, and what do you call this type of development? You call this type of development as monosporic development. One spore, megaspore, one functional megaspore is going to form the embryo sac. That is why monosporic. Monosporic means one. So megasporogenesis is the formation of megaspore from MMC. MMC's location. This is also very much important. Where do you find the megaspore mother cell? It is mostly towards the micropylar region. Towards the micropylar, where the sweater is not there, right? That is the region where you find this. That's where it undergoes division and all. So MMC division, it undergoes meiosis producing 4 megaspores, but only one megaspore remains functional and 3 of them degenerate, my dear children. And now you get the female gametophyte. But can it make the female gametophyte in a one-step process? No. There are big steps remaining still out there, and we are going to talk about that right now. So let's talk about the formation of the embryo sac. So over here I have a story again to tell. So this is the functional megaspore with its nucleus out there. You know what happens? In the case of pollen grain, what was happening? It was undergoing mitosis, right? It was undergoing forming the 2 cells out there. Similarly, here also they undergo mitosis. But the nucleus, it is not immediately surrounded by cell wall. It takes some time. So the nucleus keeps on dividing, but it is not surrounded by the cell wall. So you have to keep a hold on that. How many division takes place? There only one division takes place in the case of pollen grain. It forms 2 cells, and then one more division takes place in the genital cell, giving rise to 2 male gametes, right? That is how the process happens over there. But over here, things are quite different. So this one nucleus undergoes mitosis, the first round of mitosis. The first mitosis happens, you get 2 nucleus. Are they surrounded by the cell wall and things like that? No, not yet. Again, this cell also undergoes the second round of mitosis. This gives rise to 2 nucleus. This gives rise to 2 nucleus out there. Okay, so you have got 4 nucleuses at the end. Now again, this again undergoes the third round of mitosis. This nucleus divides into 2, this nucleus divides into 2, this divides into 2, this divides into 2. In the total, how many nucleuses do you have? Let's number it. 1, 2, 3, 4, 5, 6, 7, 8 nucleuses. How many nucleus? 8 nucleus are being formed out over here. Now, ma'am, aren't you going to form the cell wall? You said cell wall formation would be happening. Yes. At this point, my dear children, cell wall formation happens around each of them. So 1, 2, it has, so all of them, they are forming separate cells. But over here, these 2 nucleuses, I'm going to keep it like that only, because they have a separate story to say. I'm saying you females are always complicated. I'm agreeing to myself. Okay, we are complicated, and here also it is very much complicated. So nucleus which was there of the functional megaspore, it underwent 3 mitotic division. At the end, you get 8 nucleus. Out of it, almost 6 of them are surrounded by the cell membrane, cell wall and things like that. But over here, the 7th and 8th, I'm going to talk about that. Now, over here, when they form the cell membrane and stuff like that, they're going to form they're going to go into different positions, right? So, over here, you have 3 cells being formed over here. These are known as antipodal cells. So, out of them, 3 of them go at the top over here and they just settle down there. Okay, they are known as the antipodal cells. Now over here there will be two cells settling down over here, you call them as synergid cell. Okay, now one more cell comes in, that is known as the egg cell, and you know what, the egg cell is known as the female gamete. So this is the female gamete. At last we have found the female gamete out there, which is why the whole structure is known as the embryo sac. So you had the functional megaspore, it underwent division, and now when it goes again to form something like this, you call it as an embryo sac completely. Now you have the female gamete out there as well. So 6 of the cells which I told are surrounded by the cell wall and everything, they have got settled like this. Now what about both of them? These two nucleuses come over here and they are known as the two polar nuclei. What do you call it? Two polar nuclei. Ma'am, they are not surrounded by the cell wall and things like that. Basically, it becomes a part of one big cell out there. So this is one big cell out over here, and this big cell is known as the central cell, which has two nuclei which are known as the two polar nuclei. So they also get surrounded, but like this, both of them together. So in total, if you see, there is 8 nucleus for sure, but how many cells? 1, 2, 3, 4, 5, 6, and this is the 7th cell. So you have an 8 nucleate, 7 celled condition. 8 nucleate and 7 celled condition right over here. Will you remember that? And this structure, as you see, is known as the embryo sac. And if you are worried about knowing the haploid, whether it's haploid, diploid and things like that, children, my dear children, I already told you, whenever you have made a pollen grain or an embryo sac, everything is haploid only. We talk about the nucleus because since it was haploid megaspore, obviously whenever you divide, you just get, if this is N, and after mitosis it will be N only, there won't be any change, all of them would be N, 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 N. So this nucleus is having N, here also N, here also N. So all the cells, antipodal cells, they are what? Haploid in nature. If we talk about synergid, this is also haploid, this is also haploid. If we talk about the egg cell, the female gamete, it has to be haploid. When you talk about the central cell, since there are 2 nucleus and both of them are haploid, so N plus N, this was the one that becomes what? Diploid in nature. So only one cell is diploid, and that is what? The central cell. Remember that. And one very important thing about the synergids. So over here, I have to tell about their function also, right? So synergids, which you see, they have some fingery structure like this over here. It is known as the filiform apparatus. What do you call it? The filiform apparatus, my dear children. And what do you need it for? So I told you the pollen tube, it comes with the the male gametes, 2 male gametes, right? So who is the one that says that, hey, you can meet the female gamete over here? It's the filiform apparatus that would show the way. So when filiform apparatus shows the way, my dear children, it will be this, and will be carrying the male gametes out there through one of these synergids. So synergids and egg cell are kind of best friends. And synergids lays off its life for the egg cell. Basically, when the pollen tube travels through this, one of the synergid would be getting degenerated, it dies off. And that's how one male gamete will be coming and fusing with one egg cell. What about the next male gamete? I told you there are 2 male gametes. So one male gamete has come and fused with the egg cell. What about the second male gamete? So that's your homework. Put it down in the comment section. So this is homework 1. Okay, that you have to put it down in the comment section. Say that, ma'am, this is the answer. I will be really happy to see that, seeing you putting down your efforts. Okay, it's towards the next part of the chapter. We are going to cover it in the next video, but I just want you to have an understanding of things. Do something by yourself as well. Okay, so I will be obviously teaching that. So this is how the embryo sac looks, and I hope this is very much clear. Always remember 3 mitosis happens over here in the functional megaspore. Done. And now, not going ahead with the question, before that I have to complete one thing that is put over here. Where is it? Yeah. So I told you there is multiple, either you can have one ovule or multiple ovule. So whenever you see one seed, all of them will be having a single ovule. For example, wheat, paddy, mango, all of it. Papaya, watermelon, orchid, they will be having multiple ovules. Okay, multiple ovules, multiple seeds. Because ovule is the one that becomes the seed and ovary is the one that becomes the fruit later on. Okay, so that's it, my dearest little children, and now we'll jump on to the questions are there. So the question session, I have put it on different name, practice into action. Okay, practice into action session. So whatever we have studied, we are going to put it down into action. So let's begin. If you know the answer, do let me know, okay, in the comment section. So given below is a diagrammatic sketch of an anatropous ovule. What do you mean by anatropous ovule? But before that, just a second. So whatever division that I told you, the what do you call the mitosis division, what mitotic division, everything, my dear children, happens towards the micropilar end. Always remember, the micropilar end is very, very, very important. You have to always remember that. And not just that, even the egg cell is situated towards the micropilar region. The antipodal cells, they are towards the chalazal region. The egg cell will be towards the micropilar region, because that's where the pore is there, opening is there, right? So please remember that. It's not working. Okay. Oh, okay, just a second. Now, yeah. So over here, given below is a diagrammatic sketch of an anatropous ovule. Select the correct set of names of parts labeled as A, B, C, D. So here you have A, here you have B, here you have C, and here you have D. So micropyle is over here. Opposite side of micropyle is C. What do you call it? As chalaza. So you have got C, chalaza. You have three options. You can eliminate this particular option that is coming through your way. Now the next thing that you are pretty sure is basically this thing and this thing has to be integuments. This, my dear children, and this has to be integuments. Within the integuments, what do you have? You have the ovule. You would have the ovule. Inside the ovule, if at all there is something, it has to be what? The embryo sac. So D1 would be the embryo sac. D is embryo sac. You just have one option with that, and that way you form the answer also. So D is embryo sac. Outside the embryo sac, I told you the cells of the nucleus will still be persisting. So it is this. Going ahead with the next question. Generally, ovules differentiate single MMC in the dash region of the nucleus. The single MMC, single megaspore mother cell, it gets differentiated in what region of the nucleus? I told you all the important things happens towards the micropilar region. So chalazal region, micropilar region, integument region, near the hilum. What is your answer for this question? It would be micropilar region. B is your answer. So this type of questions you can think of coming in the exam as well. Children, are you enjoying the class? Are you understanding things? I hope you are. Yes. Now in microsporogenesis, the pollen mother cell, microsporogenesis, the pollen mother cell, PMC, forms microspore through mitosis, forms microspore through meiosis, undergoes mitosis and forms two similar size cells, undergoes meiosis and forms two similar size cells. Always remember, pollen mother cell, that means PMC, has to undergo meiosis and has to form 4 microspore at the end. So form microspores through meiosis. B is your answer. Cool. Do you think that whatever we learned is being put down into action? I hope so. You are feeling the same. Now the egg apparatus consists of, now this is something that you might not know. So the synergids and the egg cell together, you call the whole structure as what? The egg apparatus. Synergids, the best friends. Okay, synergids and the egg cell. So over here, the two synergids, one egg cell, two egg cell, both A and B. So both A and B can be categorized together. So this is the thing which is known as the egg apparatus. Cool. Now, going ahead with the next question. So your answer would be this, right? I hope. Yeah. Now, select the mismatched pair. Microsporangium is the pollen sac. Microsporangium is the one which is known as pollen sac because pollen grains are formed there. It is correct. Okay. Megasporangium is the ovule. That is also correct, right? Pollen grain is the male gamete. No, it is a male gametophyte. Always remember, it's not the male gamete. So this is the mismatched pair. Embryo sac is the female gametophyte. Yes, it is the female gametophyte. So what is the mismatch? C is the mismatch over here. Now, after how many divisions does a functional megaspore result into mature embryo sac? Functional megaspore, one megaspore. How many divisions are there? Mitotic divisions are there. How many of them? 3 of them. 3 of them, you get 8 nuclei at the end. So after 2 meiotic division, wrong. After 2 mitotic division, wrong. After 2 mitotic division, wrong. After 3 mitotic division, yes. So this becomes your answer. Okay, cool. Now The embryo sac is termed as monosporic development because embryo sac formation, why is it known as monosporic development? Because only one functional megaspore is the one that would be forming rise to an embryo sac. Only one, rest all of them degenerate. So their development occurs from 2 functional megaspores, wrong. The development occurs from 3 functional megaspores, wrong. The development occurs from 1 functional megaspore, yes. From megaspore mother cell, 4 linear tetrad megaspores are formed, out of which only 1 megaspore is functional. From megaspore mother cell, 4 linear tetrad, no, there is no linear tetrad being formed. This is not microsporogenesis where it is forming a tetrad of things and stuff like that. Okay, out of which only 1 megaspore is functional, this will be wrong. The development occurs from all 4 functional megaspores, wrong. This is the only answer. Their development occurs from 1 functional megaspore, straight. Okay, it's not confusing you. So this is the answer. Now there is one more question, but that's for you to solve. That is a homework question. So how many microspore mother cells are needed for the meiotic cell divisions resulting in the formation of 256 pollen grains? How many microspore mother cells do you require for forming 256 pollen grains? Your options are this. Comment down your answer in the comment section. I'll be replying to all of you and seeing whether you have got the answer. And my dear children, this is the right time to go through your NCERT. I'm pretty sure you're gonna understand each and everything about it. If at all you have any confusions left, put it down in the comment section. And if at all you're preparing for KCET, do not forget to subscribe to our channel and also click on the like button. Share with your friends who are in need. Bye bye. Take care. Lots of love. I had fun teaching you guys.